All right. Hey, everybody. If you're joining us a little early, we are just going to sit and let attendees trickle in here for the next minute or so, and we'll be on our way shortly. Thanks. I don't know. There we go. All right. I think with that, we're going to get things kicked off. Thanks, everybody, for being here, and welcome to our 22nd episode of Ewing's You. For those of you that don't know me, I'm Piero Spada, and I'll be your host today. I'm also co-founder at Little Warrior Foundation, and more importantly, father to 14-year-old little warrior Lucy. If you're part of our core contingency, aka the Little Warrior Fight Club, thanks for being here since day one with us and making this a place for positive change. If you're new, well, welcome to Ewing's You. It's our webinar series where we hear from leading sarcoma experts and discuss the cutting edge of care and science behind Ewing sarcoma. Today, we'll be hearing from one prominent expert and repeat guest in Dr. Matteo Trucco from Cleveland Clinic to discuss a new clinical trial for upfront metastatic Ewing sarcoma, otherwise known as the Mettseo trial, and what I'm calling the everything in the kitchen sink approach. A little bit, slide sharing, let's get this going. Oh, just give me 1 second. There we go. Thanks for your patience. All right, a little bit about Dr. Trucco. Dr. Trucco is currently the director of Children's Cancer Innovative Therapy Program and clinical director for the Pediatric Hemonc and Bone Marrow Transplant Department at Cleveland Clinic Children's. He is also a practicing pediatric oncologist at Cleveland Clinic Children's and Case Comprehensive Cancer Center, and an associate professor at the Cleveland Clinic Lerner College of Medicine at Case Western Reserve University School of Medicine. Dr. Trucco completed his medical training at Temple University School of Medicine in 2006, followed by residency in pediatrics at Rainbow Babies and Children's Hospital in 2009, and fellowship in Peds Hemonc at Johns Hopkins Hospital and the NIH in 2012. It was during the fellowship that Dr. Trucco started researching sarcomas in the laboratory of Dr. David Loeb, who's also been a past Ewing's U presenter. Specifically, Dr. Trucco worked on developing techniques for identifying and targeting sarcoma cells that are resistant to conventional therapy and are believed to give rise to metastases and relapses. Dr. Trucco continued his research in sarcoma resistance when he was recruited to Baylor College of Medicine, Texas Children's Hospital, and the laboratory of Dr. Jason Ewing. Here, he studied sarcoma cells' metabolism as a target for overcoming chemotherapy resistance and began his work in developing clinical trials. In 2016, he moved to the University of Miami, where he participated and developed several clinical trials aiming to overcome cancer resistance to chemotherapy and immunotherapy via the addition of multiple metabolic inhibitors and applying evolutionary principles to change how and when we administer chemotherapy to stay ahead of chemotherapy resistance. He joined Cleveland Clinic in November of 2019 and continues to develop clinical trials, both locally and in collaboration with the national consortia and pharmaceutical companies to identify new and less toxic treatments to children with cancer. In addition, Dr. Trucco has become involved in several foundations focusing on sarcomas. Dr. Trucco is also the co-chair of the National Pediatric Cancer Foundation Sunshine Project Consortium, a member of the advisory board for the MIB Agents, Sebastian Strong, Clear Cell Sarcoma Foundations, and moderates the multi-institutional virtual tumor review board, both for osteosarcoma, otherwise known as TURBO, and the virtual National Ewing Sarcoma Tumor Board, otherwise known as NEWS. For more information on that initiative, please refer back to episode number 5. But before we kick things off here with Dr. Trucco, just a quick reminder that Ewing's U is a webinar production of the Little Warrior Foundation. We are a national childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out over $2 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us at Little Warrior Foundation to make programs like this a reality, we rely on the support of the little and big warrior community alike. We appreciate all of your support so that we can continue our mission of bulldozing our way towards more effective, less toxic therapies and continue this vital work for kids battling Ewing sarcoma. With that, I'm going to hand it off to my omnipresent co-host, Emily, to introduce herself and touch on a couple of housekeeping items.
Hi, everyone. Like Piero said, I'm Emily McFadden, one of the co-founders here at Little Warrior Foundation. So glad to have all of you here. Lots of new names and loyalty, frequent flyers here at Ewing's U alike. So as a reminder, please use the Q&A function of Zoom to send questions throughout Dr. Trucco's remarks. We will save time for questions at the end. Dr. Trucco, you can ignore the questions up until the Q&A time. Piero and I will work together to field those. As a reminder, we cannot discuss any specific cases or provide direct medical advice in the context of this webinar. And before I turn things over to Dr. Trucco, I wanted to put a plug in for next week's episode. We will have Dr. Rashmi Chugh here from the University of Michigan. She is going to be talking about a phase 1 Inhibrx trial. So that is next week at this time. So on our website, littlewarriors.org/EwingsU, you can find all the information to register for next week's event as well. But with that, I will turn it over to Dr. Trucco because he's got a lot to cover today.
Thank you so much, Emily and Piero. It's always an honor to be here, and I appreciate everything you guys do for the Ewing sarcoma community. It is extremely valuable. Am I sharing my slides?
You are.
Sweet. Okay. So thank you. I'm very excited to tell you about this trial that we've developed over the past several years. Affectionately named Mettseo. No, I did not name it after myself. It was actually a colleague of ours, Damon Reed, who named it by mushing together my first name and Dr. Jonathan Metts's name down at Moffitt. And I'll kind of share how this all came about. This trial is sponsored by the National Pediatric Cancer Foundation, who's involves about 40 sites nationally of pediatric cancer centers, and has really enabled us to do some really innovative stuff. And, you know, they raise money for us, and we keep asking for more money, and they just keep raising it, and they keep challenging us to do more innovative research. And so it's been a fantastic collaboration. So once upon a time, sorry for the little issue there with the animation. So back in 2018, we had a meeting of the National Pediatric Cancer Foundation, and a paper had come out that showed 94% of all patients with metastatic fusion-positive rhabdomyosarcoma. So I'm talking about rhabdo for a second. I'll come back to Ewing's shortly. But basically, all those patients with metastatic fusion-positive rhabdo, 94% of them will relapse in 3 years. And that's despite virtually all of them, their tumor like melts away within 12 weeks of therapy. So you get this fantastic response. We're all high-fiving each other, all excited. It's great. That's at 12 weeks. You're still doing another 28 weeks of therapy because treatment for rhabdo is 40 weeks, radiation, surgery, the whole 9 yards. And yet the vast majority of those patients, almost all of them, it's coming back within 3 years. The other 6% hopefully are cured. Maybe their disease will come back after 4 years. So what does that mean? And when looking at it, we were kind of thinking, it's prompted a discussion within our consortium about maybe we got to look at these cancers in a different way, because that behavior gets back to what Piero mentioned I was doing research on in the past, these kind of stem cells that resist all the therapy you give. You can't see them because there are very few of them, but then they give rise to a new tumor. And this prompted a discussion, especially because MPCF is based in Tampa and associated with Moffitt Cancer Center, or works through Moffitt Cancer Center. And they have a department of evolutionary biology and mathematics at a cancer center, which seems odd. And so that started this conversation of how might we borrow some features of evolution and apply it to cancer, because essentially we're trying to make cancer extinct. But if you're not able to, for some reason, maybe we can borrow some ideas from evolution. And so that led to this trial, which is called Evolutionary Therapy in Rhabdomyosarcoma. Again, it started, I believe, in 2020. Yeah, we actually here at Cleveland Clinic enrolled the first patient. The fact that it went from 2018 to 2020, from idea to enrolling the first patient, that is lightning speed. So we were talking with Piero before the webinar started. Still not fast enough, but 2 years it's ridiculously fast for clinical trials. And that was kind of sticking our toe into the water. Basically, we're testing a couple different ideas of, if you hit it with more chemo, that doesn't seem to be the answer. Just so maybe we got to come at it a little bit different. And so this is a Ewing's talk. So what came of that? So in Ewing sarcoma, there was a paper by Landenstein and colleagues back in 2010. They were trying to figure out how can we prognosticate, predict who's going to do well and who's going to do poorly in Ewing sarcoma. And these are sobering numbers. But basically, if you get a score and it's based on the patient's age, where their tumor is, where it's metastasized, the size of the tumor, you get a score. And you can see here, if you have a score of more than 5, your 3-year event-free survival, again, it's terrible. It's 8%. That means only 8% of patients will be disease-free at 3 years. Again, these are patients who have metastatic disease as diagnosis. It's beyond the lung. It's in the bones. It's in the bone marrow. And again, it means they're going to be relapsing by 3 years. Now, don't ask me why they messed up the legend here, but again, this is the line that has the highest risk of it coming back is down here, and it's the higher score. And we know from other studies, this one's from Greer back in 2003, where they added ifosfamide etoposide to the standard chemotherapy at the time, which was vincristine, doxorubicin, cyclophosphamide, so the VDC. And this was the standard for localized disease, the blue, and adding the ifosfamide etoposide, the IE, definitely improved outcomes. And that's why the standard is now VDC-IE. For metastatic disease, though, the standard was the red line, and adding the ifosfamide etoposide did absolutely nothing. And the question is, well, why do we add IE even if you're a metastatic patient? Excellent question. A lot of debates gone on about that. The idea is we don't want to be less aggressive to the more aggressive disease. Maybe some people might benefit from it. Maybe it helps space things out. But those of us who kind of do this a lot, we know that this is the data. And if a patient's having trouble with ifosfamide etoposide, we are quick to drop it because in the upfront setting, it doesn't really yield any benefits. So this is showing us that adding drugs that we know have activity against Ewing sarcoma don't change things for metastatic patients. So clearly more chemo is not the answer. We got to come up with a better idea. So as we were getting that rhabdo trial up and running, Damon Reed, who was at Moffitt and now is the head of pediatric solid tumors at Sloan Kettering, he was running the Sunshine Consortium. Me and Jonathan were his vice chairs. it was in the midst of COVID. We didn't know what was going on. And so in that kind of brief pause where everything was going virtual and we were figuring out what life was going to look like, he was like, hey, here's a thought experiment. We got this rhabdo trial. We're going to be getting it up and running. What would we do for metastatic Ewings? We have those Landenstein scores and the prognosis. That's terrible. How would we design a Ewing's trial that really took a deep dive into this evolutionary theory of how we might get rid of Ewing sarcoma, metastatic Ewing sarcoma? And so, first of all, we came up with some guiding principles. And to be honest, we actually had to come back to these multiple times because as we were discussing it, we kind of broadened the discussion with the rest of the consortium. You know, we started getting way off track and we kind of had to rein it back in. So there are a couple principles. One is we're not one drug away on metastatic sarcoma. We're not even 2 drugs away, which, you know, adding ifosfamide etoposide showed us right there. The most recent metastatic Ewing's trial was with ganitumab. That's the the GD2 antibody, the insulin-like growth factor receptor antibody, and compressed VDCIE chemo, and that didn't show any improvement. So the idea we wanted to test was whether what we call sequential second strikes was helpful, and that's alternating the therapy regularly and frequently to try to stay ahead of relapse. We want to incorporate maintenance, which is borrowed from pediatric leukemia, where it's a kind of a low, steady chemotherapy for a prolonged period of time to grind out any cancer cells that might be still around. We didn't want to, we wanted to test the strategy, not new drugs. Absolutely, we hope there are new drugs. We're excited about new drugs, but we wanted to see, would changing the strategy using drugs that we already know are active in Ewing sarcoma improve the outcomes? So again, we're testing the new application and schedule of existing drugs that we actually know have some activity in Ewing's. We wanted to maximize the days or weeks of receiving therapy, including extending the length of treatment, but do it in a way that's tolerable. We wanted to incorporate biology and kind of study things as we're going along during the trial so that when we get the results back, we can, whether it's successful or not, we can hopefully learn as much as possible to inform the next trial. And then I know Piero used the kitchen sink, and we actually did a kitchen sink trial for rhabdo called ARST0431, and that was no improvement. So we wanted to avoid the pitfalls of that, and hopefully we did. So this is Ewing sarcoma therapy, and I made it small on purpose, but it's, you know, VDC, i.e. back and forth, back and forth, back and forth, surgery, radiation, then back and forth, VDC, i.e. This is what they do in Europe, where they use VIDE over and over and over again, surgery, radiation, VAI, and then they actually do, on this trial, high-dose chemo with stem cell rescue, so like bone marrow transplant for it. This is that kitchen sink. So while they threw a lot of active drugs, you kind of see it's still somewhat monotonous or repetitive. This is a completely different tumor, neuroblastoma, where we have slightly better outcomes. The survival for metastatic neuroblastoma, high-risk neuroblastoma, is about 50%. And there they use, you know, this is cisplatin, topotecan, ifosfamide etoposide, VDC. You can start seeing they switch things up: surgery, radiation. They do high-dose chemo, stem cell rescue. Then they got these maintenance phase with retinoic acid, and they have some immunotherapy thrown in there. So again, a lot more different approaches. This is leukemia, standard risk leukemia, which is like the poster child of pediatric cancer. It's the most common cancer we see. went from universally fatal in the 1950s to now we cure 85, 90% of patients. And again, this kind of, you start with one thing, then you switch, and then you switch, and then you maybe go back to the first thing, but then you switch again. And then you got this prolonged maintenance phase. And there's a lot of drugs built in there. So we wanted to take our standard approach to Ewing sarcoma and make it approximate something like this, because as you go down this list, the outcomes improve. So, and we've written a couple papers about this. This is, just basic for all sarcomas. Then we wrote this specifically about our approach for Ewing sarcoma. And then most recently, we published this one really borrowing from leukemia of what they do. So again, different tumor, but in leukemia, patients come in with massive amounts of leukemia cells floating around. And their bone marrow is chock full of leukemia. And in 1 month, they get either 3 or 4 drugs. And at the end of that 1 month, we do a bone marrow, and we want to see that there's virtually no leukemia left in that bone marrow. And if you responded well, that's what we find. You think, fantastic. You went from your marrow is 100% leukemia to no leukemia. What are we going to do? We're going to give you something completely different. That's how leukemia therapy happened to come about. It wasn't necessarily intentional. They weren't purposely switching, but you get hit with something completely different drugs. And then you'll come back to some of the drugs you saw before, but you go into a less intense phase, let the patient recover a little bit. Then you hit them with something intense again. It's called delayed intensification. Then you have another little gap. And then you have this long, drawn-out maintenance phase, which is like 2 years, that they're on low, slow chemotherapy. And so we wanted to see maybe that's a better approach, because if you're talking to an evolutionary biologist, they hear what how a leukemia trial is, and they said, that sounds like background extinction. And it's like, well, what's background extinction? We all ask them when they talk to them. And it's the extinction we see in most, species that went extinct. We're not talking about, giant meteor hitting the earth, wiping out all the dinosaurs, even though even that, it's not one impact. It's the fallout of that impact that we assume wiped out the dinosaurs. But background extinction is lots of little things that are seemingly unrelated that built upon each other to eventually wipe out species. And I'll go into that a little bit more. But, in February, we started talking about evolutionary approaches. We opened the rhabdo trial. We started having this idea of how to apply this to other sarcomas. We decided we'd focus on Ewing's. We presented the idea for the first time in 2021, February 2021, and no one said no. We were throwing a bunch of stuff. We had transplants in there. We were just, seeing what would stick. Incorporated a bunch of feedback, revised it. In 2021, we presented it to the group again, got even more feedback. We even presented it to COG and got some feedback from them. And again, kept working on it. 2021 was a busy year. And then we were trying to figure out, some of the biological correlates we could do along the way. Eventually got it all on paper, got it reviewed, got it approved. And right now we are eminently ready to open it. We're just finalizing some budget issues. And then we should be ready to open it. So it's gotten all the approvals. We're just dotting some I's, crossing some T's. And again, it wasn't just me. It wasn't just me, Jonathan, and Damon. It was a team effort to get us this far. So background extinction. This is a passenger pigeon who, not in our lifetime, but in recorded history, went from being so abundant that they blacked out the sky to being completely extinct. And what caused that? Well, first, hunting. People would hunt them for game, for food, but that wasn't enough to wipe them out. Habitat loss, with the Industrial Revolution and all that, deforestation, that had an impact. Then there were some outbreaks of infections among the populations. Then there was some food scarcity, then a particularly bad winter, and just kind of dwindled and isolated all the populations to the point where there was only one bird left named Martha, and she was in the Cincinnati Zoo, and she passed away in, I believe it was 1914. And so in a very brief period of time, we actually saw a series of events that led to the extinction of a population. And that's what background extinction is. And that's what we're trying to model in this approach in knocking out metastatic Ewing sarcoma. So this is what it looks like in broad terms. You get diagnosed with widely metastatic Ewing sarcoma. So this isn't just localized disease. It's not just localized disease with like one pulmonary metastasis. We're talking about you have Ewings in your bones and your lungs and your marrow, like all over the body. And they're fortunately rare, but these are the worst of the worst. Again, usually they respond to therapy pretty well, but it comes back pretty quickly. So we're not throwing the baby out with the bathwater. We are using VDC because we know that works and we know it responds. But after 4 cycles, we're switching to something else. And this is called Iriva. It's irinotecan, ifosfamide, vincristine, actinomycin. Again, agents that we know have activity. The timing here is different. You get 3 of them on day 1, and then on day 4, which is kind of a weird day to give it, you hit them with irinotecan, specifically to try to catch the cells that were not actively dividing on day 1. Maybe they're actively dividing on day 4, and that's when you're hitting them. So it's kind of, again, trying to change the scheduling, the strategy. Then we give the patient a little bit of a break because those are some pretty intensive therapies, and we just go to a TKI. Cabozantinib is the one we picked. We actually, incorporating feedback, people were concerned they wouldn't be able to get access to cabozantinib, so we built in some alternatives, but along with radiation to the primary site, so we don't have to interrupt therapy for surgery. Again, rarely do you actually propose surgery for someone who has widely metastatic disease. Fortunately, radiation can help. Incorporating feedback from physicians and some patients, was like, well, what if surgery was the best option? So we said, okay, if surgery is the best option, you can do surgery here, but we're gonna, skip the cabozantinib because it interferes with healing. And once you've recovered from that, you will jump right into cyclotopo. Cyclotopo and high-dose ifos are some of the most active agents in relapsed Ewing sarcoma. So we're incorporating them, again, 2 cycles, just to kind of hit them hard, hit them fast, switch, hit them hard, hit them fast with something else. And these kind of line up with the phases in leukemia treatment. Then we address the metastatic sites with more radiation and irinotecan and temozolomide, another very active, relapsed Ewing's regimen. And temozolomide and irinotecan are safe with radiation, and in fact, the temozolomide helps the radiation work. And then we have this prolonged maintenance phase, which is, again, trying to make it tolerable. It's combining oral drugs and drugs that you only get like once a month. And then we built in, biopsies of the tumor, drawing blood. We went back and forth a bit of whether or not we could make biopsies mandatory, especially for the younger kids. It was deemed no. But if for whatever reason a patient is getting a biopsy because we want to see whether it's responding, or for some other reason, or they have bone marrow disease, which you actually have to check to see if you clear it, we will collect those samples along the way. And then we'll collect the blood for circulating tumor cells and circulating tumor DNA. We include standard of care imaging, but also some research imaging to see if we can kind of use that to help build models and predict the outcomes later on. So that's the general scheme and the why. It is for all newly diagnosed, widely metastatic Ewing sarcoma. Actually, it's not family tumors, just Ewing sarcoma. That means metastases beyond the lungs. If you have a tumor in your leg, and then you have a met to one of the other bones and bone marrow involvement, that counts, because those are the ones that do worse. The reason we don't include the ones with localized disease or those with metastases just to the lungs is because all in all, they actually do relatively well with the standard of care, and we didn't think it made sense, to give someone completely experimental therapy when they might have, 50 or better chance of being cured with the standard of care. Again, we built it using radiation for primary and metastatic site control, but you can get permission to do surgery instead. And the patient needs to be willing and capable of complying with these regimens and evaluations. Again, keeping it very loose. If you have localized or lung-only metastases, you can't enroll in this. There will be a COG trial coming out soon that actually is available for lung-only metastases. So it's not like those patients are without options. This is not for relapsed refractory disease. This is testing a theory up front to try to stay ahead of the resistance. If you've already relapsed or had refractory disease, kind of in a way, I don't think this strategy is the best approach. Yes, we need things for those patients and we're working on it, but we wanted to kind of see if this worked in that upfront setting. And if you can't kind of follow the protocol, then obviously you should not be on the protocol. So this is what it looks like with each individual drug. And then, as you can see, we kind of use some of the same ones, mix them up. There's some that were there before, then you introduce them later, then you introduce some new drugs. In this maintenance phase, we actually have oral drugs, oral cyclophosphamide and oral etoposide for 1 month, and then alternating with months where you just get vincristine and liposomal doxorubicin on day 1 of a month, and you go back and forth. So again, that hopefully is relatively easy for patients to manage and deal with. It certainly doesn't mean you're coming back to the hospital every single day. It should be stuff that you can do at home. Compare that to the current standard, and again, hopefully you see that there's a lot more diversity. It's stretched out, and honestly, a lot of these regimens can be given outpatient, and I'll show you on a future slide, you're actually getting less chemo or comparable chemo, but stretched out over a longer period of time. And this is kind of what leukemia looks like. And again, sort of trying to map it where you're getting this induction, you're switching, you get consolidation, you get this intermaintenance, which is kind of a lesser chemo for a little bit. Then you get this intense period of delayed intensification, then another little interim maintenance, a milder period, and then you get into that maintenance phase. Again, not all the drugs that work for leukemia work for Ewing sarcoma, so we can't model it exactly. I wouldn't treat someone with Ewing sarcoma with leukemia therapy, but more that strategy and that kind of the various phases of intense, less intense, switching up the therapies along the way to kind of try to avoid resistance developing. This is the cumulative doses, how much chemo you end up getting. With the standard of care, you're getting 8.4 grams of cyclophosphamide, 64 grams of ifosfamide, 375 of doxorubicin, and 3,500 of etoposide. With our Mettseo, again, over a much longer period of time, you get a little more cyclophosphamide, but actually less ifosfamide, a little bit more doxorubicin, but it's given as liposomal doxorubicin for a big chunk of it. And then a little bit less etoposide, and it's actually all given orally. So again, not saying this is mild by any stretch of the imagination, but it's not like we're being more intensive. We're just trying to be a little bit smarter in how we're doing it. And when you kind of compare alkylators, which cyclophosphamide and ifosfamide are, you're actually getting less and those have a big impact on fertility. Again, I think 50 grams will still probably impact someone's fertility, but generally speaking, we're overall less. So again, alternating periods of intense and less intense, changing it up. Most of these things can be done outpatient. The VDC, we do it outpatient sometimes. IRIVA probably requires you to be in the hospital. Radiation with cabozantinib can be done outpatient. Topocyclo could probably be done outpatient. High-dose ifosfamide, we do a continuous infusion, so can be done outpatient, but usually this is inpatient. Temo, outpatient, and these maintenance are intended to all be done as outpatient. And again, evaluations, blood samples all along the way. Yeah. So again, a huge thank you to the National Pediatric Cancer Foundation for supporting this and funding it. Again, Damon Reed challenging us to come up with, some ideas how we would tackle it. Jonathan Metz has been my partner in crime for this whole endeavor. Tom Russell brought some leukemia expertise to the early discussions. The protocol committee at MPCF, Mike Isakoff, Lars Wagner, Scott Bornstein, Jonathan Gill, Bhuvan Assetti, David Loeb, Leo Mascarenas, Rosina Pertilis, Ajay Gupta, Michael Dietz, Mas Hayashi, all of them and many more contributing ideas and thoughts. The front staff, as we call them at NPCF, have been incredibly helpful in like reading over our protocols to make sure it made sense. All the sites that have supported this, we had a up-down vote when it was still just an idea, and 88% of the people in attendance voted to move this forward. The two that actually opposed it said, just because I don't think I can get those drugs. And so we built in alternatives if necessary. Pete has weighed in and seems very excited about this idea. Children's Oncology Group, they said we probably can't open that in the Children's Oncology Group right now, but do the trial as it's a small trial. It's just like 20 some odd patients to see if we can, if there's a signal. And if we have a signal, then they're happy to have us bring the idea and the data back to then open a larger trial to really see if it works. Little Warriors has been supportive all throughout. I've shared, the papers and the ideas along the way. And all the patients and families that we've actually talked to is like, would this be something that you'd even consider? And, when you look at the numbers, a lot of them would be. I'm not saying it's for everybody, but a lot of families, when they hear the numbers and see the numbers, they're like, we need something different. And again, we have the patient in mind. We want to cure them and do it in a way that's tolerable. And so hopefully this is it. With that, I'm going to stop talking and answer any questions you guys might have.
Thank you so much, Dr. Trucco. That was great. And I made a note, you had a bullet point that said conversations with anyone that will listen. And I feel like that's a guiding principle of the Little Warrior Foundation too. So really, really enjoyed that. We did have a few questions come in, and I know even more will be coming through. So you shared the European approach. How does their survival rate compare to the US?
Honestly, about the same. That Landenstein, they're I'm gonna get in trouble. They're from Europe. That paper's from Europe, so those outcomes are comparable. And in fact, there's still a lot of debate whether or not VDCIE versus VITA plus transplant is the way to go. There's a lot of controversy in whether or not bone marrow transplant really has a role in Ewing sarcoma. And it's not that there aren't some patients that do well after bone marrow transplant. It's that those are the patients that did well with chemo, usually. You're not going to take someone to transplant who progresses on chemotherapy. So there's this concern that there might be what's called selection bias. You are transplanting the ones that are going to do better anyway. And so, of course, it makes it look like the ones that transplanted did okay or better. There's also a lot of toxicity involved with transplant. So while in some studies there might look like there's a benefit in survival from the cancer, you have to balance that with the mortality from the actual treatment, which is not zero. So again, the Europeans aren't performing much better than us, unfortunately.
Cool.
Wish they did. That way I'd steal their ideas. But
Absolutely.
Yeah.
All right. Thanks for that clarification. And I apologize on my end for labeling and mislabeling this trial as a kitchen sink approach. That wasn't my intent. It was just basically just to highlight the diversity in the array of number of therapies going into this. I know there's method to the madness, and I'm glad you clarified that for us all.
Well, and I will say one thing that I meant to show it when I had the comparison, but imagine you do your 28 or 29 weeks of standard Ewing sarcoma therapy, and then 3-6 months later, a patient relapses. Well, we're going to grab the high-dose ifosfamide or the vincristine irinotecan. They're going to get that. And that might work for a little bit, but then they'll progress or relapse. And so we'll throw topocyclo. And then so we're basically giving those same drugs. It's just instead of giving it a chance to become resistant and reemerge, it's like, let's just hit it.
Right.
And hopefully by hitting it and not taking the foot off the gas, we get rid of it once and for all.
Yeah, no, the approach is sane for sure. So there's a question here from, I'm assuming this is cumulative dose. What is the lifetime dose of doxorubicin a patient can receive? And I'm assuming it's based on toxicity. They thought that the current dose was all given up front, i.e. you couldn't have any more doxorubicin after upfront therapy. Can you comment on that?
Yeah. So back in the day, we didn't have dexrazoxane, which is a heart protectant. So we would get close to 400, and that was kind of where we would draw the line. And that's why VDCIE is built the way it is, for osteosarcoma MAP is built the way it is. So since then, there's a couple things you can do. One is if you've been giving dexrazoxane protecting the heart with every doxorubicin dose, we've actually been able to push the doses higher. Again, we still check echoes and all that, but Dr. Anderson and I were commenting that we haven't seen heart failure in a while, ever since dexrazoxane was developed. So that lets us push the doxorubicin dose higher if we want. Doxil, liposomal doxorubicin, it's the same drug, it's just packaged differently, and it also doesn't have toxic effects on the heart, or at least doesn't seem to have the same degree of toxic effects on the heart. We have given some patients over 1,000 milligrams per meter squared of Doxil, and they've been okay. Makes us nervous. Follow them like a hawk. We do an echo with every dose, but we've been able to push that dose a lot higher. So I think it came out to 650. I'm not worried about 650 if we're doing it with dexrazoxane and we're doing it with liposomal doxorubicin, with the understanding we're doing echoes to check.
Got it. Thank you.
Thanks. Can you talk a little bit about what life looks like on this trial? I know it's extended length, but a lot more opportunities for outpatient treatment. But are kids expected to be hit and down and out so hard in this, or should it be a more palatable day-to-day life?
Yeah, I mean, it's a little hard to predict, but here, let me share my screen again. Okay. You guys see the comparison?
Yep.
Okay. So, there we go. So this is your standard Ewing's therapy, and you're, up to here, and you're coming into the hospital every other week.
Mm-hmm.
So yes, these first 14 weeks you're in the hospital kind of every other week. These like week 10 and 11 you're not in the hospital. You can get those outpatient. This whole primary site radiation can be done outpatient. Again, you have a couple weeks off in between these. The one concern is that this is high dose ifosfamide over here, these blue ones, and that's towards the end of therapy. And there's a little concern of, how rough is that going to be when you've gotten, 27 weeks of chemo up until then? Is it going to take you a long time to recover? Absolutely valid. We were intentional in having it later, and hopefully, since up until then things are not as bad as your standard VDC, i.e., hopefully you'll have more reserves then. But after that, after week 32, this is all outpatient.
Mm-hmm.
It should be like, this is the stuff we do in like a multiply relapsed patient who's just wants something. Their marrow's been beat up and they want to focus on quality of life. I mean, yeah, I'll do some oral cyclophosphamide and oral etoposide. Yeah, I'll do Doxil. So again, everything past week 32 should be outpatient, should be just minimally toxic. Even the oral things, we kind of plan on checking. And if your blood counts drop too much, well, we're giving you too much drug. So we're going to kind of like titrate it to try to keep their blood levels in a safe place, so they're not getting, neutropenic fevers or anything like that. Again, it's trying to slow and steady grind out any cells that might still be around.
Great, thanks.
All right, we got another question here that's talking about, since this is a multimodality therapy, and if you do start to see improvement in OS or EFS, how are you going to be able to pinpoint what is, the agent that's most responsible for that? Is that where the tumor biopsies and all the blood samples come in to play? Can you speak to that a little bit?
Yeah, so, I had some of the other slides with the arrows, and, we're hoping either with liquid biopsies, blood samples, or tumor biopsies, or the scans, it'll clue us in a little bit. There's also a lot of mathematical modeling that happens around that, which again, we can only do after we get all the data and then plug it into, some models to try to predict. Truth be told, this is totally made up in that sense. We have no evidence to say we should do this after this after this. So yeah, if it responds and we see improved outcomes, that's great. What was it? Honestly, that's a good problem to have, is
Right.
kind of how we're looking at it.
Right.
And then we can spend the rest of our lives trying to figure out why, 50, 80, however many % of patients are cured by this. Let's just get there first. But it's all those biopsies and scans and everything play into that. Hopefully, again, this is a small study. It's not going to answer anything definitively, but if there's a little bit of signal of some improvement, then we go to COG, we do the bigger study, and maybe there we collect a little more tumor samples or scans to try to really answer that question and see if we can optimize that combination or sequence.
Like you said, that would be a good problem to have, right? To try to figure that out.
Yep.
All right, we have a question here. Has data come out about the effectiveness of the rhabdo evolutionary trial? Curious as to the impact this approach has in another solid tumor.
Yeah. So for the rhabdo one, again, was just sticking our toe into the evolutionary pool, if you will. So rhabdo is 40 weeks of what we call VAC, vincristine, actinomycin, cyclophosphamide. You just do it over and over every 3 weeks. The first arm of that was, maybe we need to be more intense. We need a bigger meteor, so to speak, to make things extinct. That's called first strike. And so we took the vincristine out and we put vinoralbine, which is a stronger agent, in that, and tried. The second arm was, we're going to do VAC because VAC works. It makes the tumor go away, but then we're going to switch before 40 weeks. Why are we doing the full 40 weeks? Let's switch to something else sooner to see if maybe we can still have the same control, but maybe less toxicity. So kind of a maintenance phase incorporated earlier. And again, you've gotten rid of the tumor that you can see and switched to something else that might help keep it away. Those were the first two arms. The third arm was the really out there approach, and that was borrowing from a principle from actually farming. So if you have a farm and you have a crop and you have a pest, if you bombard your crops with pesticide, yeah, you'll drive down the population of pests, except eventually they'll become resistant and the resistant population will grow and eventually start affecting your crop again. You hit it with another pesticide and you drive it down, and then a little bit will be resistant and they'll grow and eventually affect your crop again. And you do that over and over, and all you do is develop really resistant pests, douse your crops with every pesticide known to man, and you've kind of, blown a bunch of money. Farmers don't do that anymore. They learned their lesson. What they do is they spray just enough pesticide to bring down the population of the pests so that they're not affecting the crop. Then they take it away and let the population grow again, because the population that will grow is the sensitive population. If they were sensitive in the first place, that's the dominant population. And then when they start affecting your crop again, you hit them with another dose, drive them down, take it away, let it grow back. Hit it, drive it down, take it away. And that way you can keep the pest population under control, doesn't affect your crop. You spend a lot less money on pesticides. You expose your crop to a lot less pesticide. Resistance doesn't develop. They did something like that in prostate cancer, and it actually seemed to get control of metastatic prostate cancer in old men using smaller doses or less chemo over longer periods of time, and they remain sensitive. So we'd kind of give the chemo, we'd measure PSA, which is a blood test for prostate cancer, drive it down to 50%, take the chemo away until the PSA gets back to baseline levels, give the drug again, drives it down, 50%, take it away, wait until it comes back up. And again, a lot more patients were alive longer with less drug, with less side effects, using that give, take away approach. So we were trying to do that with VAC chemotherapy for rhabdo. Truth be told, only one patient went on that arm. It is a hard sell saying your child has cancer, we're going to give them chemo, and then we're going to take it away and let the cancer come back, and then we'll give it again. So an adult patient actually enrolled in that, did relatively well. It is, despite all that, a little too early to tell you the results. Were there some patients that did better than expected, compared to VAC? Yes. Were there some patients that did probably worse? Yes. It's just too early to say. But we have enrolled in a lot of almost all of those arms. And as soon as we have all the data, we'll share it. But again, that's like one little step compared to what we're doing with this trial.
Yeah. So too early to tell on that one for sure. Yeah. Bringing me back to my integrated pest management courses back in the day, talking about crops.
There you go.
Anyways, keep us on topic here. There's another question about doxorubicin and toxicity, heart toxicity. Is it more acute toxicity or long-term toxicity that we're concerned about, especially if we're pushing the upper limit? Can you comment on that at all?
It's when you start going above 400 milligrams per meter squared without cardioprotection, you see heart failure. Some of that happens like right away. Like you get an echo, and instead of having an ejection fraction of 66%, which is about normal, it drops to 40. Well, then you have an acute toxicity, and that's not good. Again, we're not seeing that anymore as much as we used to. Okay. The long-term toxicity, also, we have had plenty of patients where we gave all the doxorubicin, they were fine, again, without cardioprotection. And 5-10 years later, they show up and, oh my goodness, they're in heart failure. And so it's both. With the dextrazoxane, we aren't seeing the acute toxicity. We're starting to get into the period of patients had been treated with dextrazoxane for all of their doxorubicin, and it's been 5 years, 10 years now. And again, we're not seeing that cardiotoxicity that we used to see. So I think it helps for both the cardio protection, but it might be a little early for the long-term toxicity.
Gotcha. Thanks.
That catches us up on our questions. You do have some fan mail from our celebrity guest. Dr.
Oh my.
Peterson is here. He said 2 thumbs up. So that's high praise from Dr. Pete.
Thank you, Pete.
I had one kind of final question of just about, we are obviously hoping and praying for the wild success of this trial. You said, COG might be interested once they start to see results. Could you just put in our heads kind of the timeline of, when, how long would an organization like COG be following this before they would feel, is this like 3 years, 5 years? How long do they need in order to feel comfortable if we're starting to see strong results?
Sure. So remember that I said something like only 8% of patients are disease-free at 3 years. Yeah.
Mm-hmm.
So again, we need to go out 3 years on this trial to see, okay, are we better than 8 or not? So the trial itself is going to take several years, one, to enroll patients, because again, fortunately, it's a small percentage of Ewing sarcoma patients that have widely metastatic disease. So to enroll, and again, hopefully all of them enroll in this trial, but we might not get all of them. So it's only a few a year. 2, we have to go through the whole treatment. 3, we got to follow them for 3 years to see if they really stay disease-free. So it's going to be several years before we actually have the data to say, yes, this is really working. And then COG, because you're now dealing with, instead of 40 sites, 200 plus sites, and the federal government, again, it just takes a little bit longer to enroll to open a study. Once you open it, they enroll like gangbusters because again, it's 200 sites. And again, we might, if it's really promising, we might say, okay, listen, all metastatic disease, it might be.
I guess to follow up on that, I may have missed that, but for this trial, is it only opening at Cleveland Clinic or starting at Cleveland Clinic and then, sorry, go ahead.
So it's actually going to start at Moffitt because it's through the National Pediatric Cancer Foundation, which, because they're based in Tampa, they kind of use the infrastructure of Moffitt. But it's 40, our consortium is 40 sites. I don't think all the sites are going to open it, but hopefully many are going to open it. So hopefully Cleveland Clinic is one of the first ones to open it. Be a little embarrassed if it wasn't.
Right.
But, and again, our consortium, it started in Florida, so obviously there's a preponderance of sites in Florida, but then it kind of has grown nationally. We have Seattle, so there you go, coast to coast.
No sunshine there, but
yeah, absolutely.
We bring a little sunshine.
You bring the sunshine, absolutely. And the date that it's going to open, any day now, right? TBD.
Yeah.
That's what everyone's going to ask us. Yeah.
Exactly.
Okay. All right. With that, we'll bring this session to a close. Dr. Trucco, thank you for your time today and for sharing this clinical trials. Highly innovative, outside the box, obviously an unmet need and an enormous amount of work that you and your team put together and putting this together. So thank you for sharing your time with us and sharing this trial with us. Thank you to my fabulous co-host, Emily, for being here and helping me moderate conversation as always. Lastly, thank you to our viewers for joining us today as well and on this fascinating discussion. Please remember to stay up to date on future Ewing's U episodes. The easiest way to do that is subscribe to our Ewing's U emails. You can do that by going to littlewarrior.org and navigating to our Ewing's U tab on our website. Also follow us on social media, Instagram and Facebook, as we always post about our next speaker that way as well. And as Emily already alluded to, our next speaker is already scheduled. same time, same place. So Tuesday, next week, Tuesday, April 22nd, 1 p.m. Eastern, we're going to be meeting with Dr. Rashmi Chug to discuss the new trial centered around Inhibrx 109. So targeted agent for relapse disease. Hope to see you there. And that's it for now. See you next time. Swords up, everyone.
Swords up.
Thank you.
Thank you.
Bye.